Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic Human Genome Project. So it is also called HGP. So this is from the chapter Molecular Basis of Inheritance. So watch the entire lecture to gain basics out of it. So without any delay, let's start with the topic Human Genome Project. So let's start. So let's start with the topic Human Genome Project. In short form it is called HGP. So what is Human Genome Project? We'll see that there are numerous chromosomes, and inside the numerous chromosomes there are numerous genes are there. We know that we have 46 chromosomes, that is 23 pairs. So we'll know the function of each. Okay, so the entire DNA in a haploid set of chromosome of an organism is called genome. So what is a genome? The entire DNA in the haploid set of chromosome of an organism is called a genome. So in a human genome, DNA is packed in 23 chromosomes. Okay, so in human genome, DNA is packed in 23 chromosomes, and human genome contains about 3 into 10 to the power 9 base pair. Human Genome Project, it was started in 1990 and it was ended in 2003, and it was the first mega project for the sequencing of nucleotides and mapping of all the genes in a human genome. Okay, so it was a very big project, and here in this project we could map the different genes in a chromosome. Okay, and it was studied for human being, and Human Genome Project was coordinated by US Department of Energy and National Institute of Health. So we know that we have 46 chromosome, that is 23 pair. Inside the chromosome we have genes, so we have to study, and in this project they studied the functions of genes. Now let's see what are the goals of Human Genome Project. So you can see first one is to identify all the estimated genes in human DNA. First we have to identify all the estimated genes in human DNA. Next, sequencing of 3 million, okay, sequencing of 3 billion chemical base pairs of human DNA. First one is to identify all the estimated genes in human DNA, how many genes are there. Second, we have to sequence 3 billion chemical base pairs of human DNA. Next, we have to store this information in a database. Fourth, we have to improve tools for data analysis. Next, we have to transfer, okay, so transfer related technologies to other sectors. And last is address the ethical, legal and social issues, that is ELSI, that may arise from this project. Okay, so these are the goals of HGP. So you can see left hand side the structure is given, one chromosome, first a cell is given, inside that chromosome, inside the chromosome DNA. Okay, and inside the DNA we have genes, and below you can see the nucleotides. Next, in Human Genome Project there are two methodologies. First, it involves you can see it involves two major approaches. Okay, so under methodologies of Human Genome Project, it involves two major approaches. First one is expressed sequence tag, that is EST, and second one is sequence annotation. So express sequence tag, you can see it focused on identifying all the genes that are expressed as RNA. So first one, it expresses or it focuses, okay, so it focuses on identifying all the genes that are expressed as RNA. Second one is sequence annotation, that means sequencing whole set of genome containing all the coding and uncoding sequence, and later assigning different regions in sequence with functions. So in the right hand side you can see the pictures. Now what is the procedure of Human Genome Project? First is to isolate DNA from a cell. Okay, so the procedure of HGP is we have to isolate DNA from a cell, then we have to convert it into random fragments. Next, we have to clone. Okay, clone in a host bacteria or yeast. Okay, we have to clone it in a host bacteria and yeast using vectors like a BAC and YAC. BAC means bacterial artificial chromosome, and yeast artificial chromosome for amplification. Okay, next is sequencing of fragments using automated DNA sequencer using Frederick Sanger's method. Next is arrange the sequences based on overlapping regions. Next is alignment of sequences using computer programs. So you can see BAC is bacterial artificial chromosome and YAC is yeast artificial chromosome. So you can see the pictures in the right hand side. First is we have to isolate the DNA, then we have to convert it into fragments. Okay, next we have to clone it. Okay, we have to clone it in a host, and we have to use vectors for amplification, and then we are using the automated DNA sequencer. Then we have to arrange the sequences, and then we have to align the sequences using computer programs. So this is the procedure of human project. You can see the pictures are given. Now this is Frederick Sanger. The picture is given left hand side. So Sanger has also developed method for sequencing of amino acid in proteins. So DNA is converted to random fragments as there are technical limitations. Okay, there are technical limitations in sequencing very long pieces of DNA. So HGP was closely associated with bioinformatics. Okay, so HGP was closely associated with bioinformatics. Bioinformatics means application of computer science and information technology to the field of biology and medicine. So what is bioinformatics? It is the application of computer science and information technology to the field of biology and medicine. So next you can see out of the 24 chromosomes, okay, there will be out of 24 chromosomes, that is 22 autosomes and one Y and X. The last sequence one is chromosome 1. That has started in May 2006. Okay, not 206, it's 2006. So genetic and physical map of genome were generated using information on polymorphism of restriction endonuclease, a recognition site, and some repetitive DNA sequence. These are called microsatellites. So what are microsatellites? They are repetitive DNA sequence. Okay, so your restriction endonuclease is used, repetitive sequence DNA sequence are used. So DNA sequencing also have been done in bacteria and yeast, okay, as well as in Drosophila plants. All right, so there will be total 46 chromosome. Out of that, 23 pairs are there. Okay, so you can see in the center, that is the 20 out of the 23 pairs, 22 pairs are same for male and female. Only the 23rd pair you can see, and that is XX is for female and XY is for male. Now let's see the silent features of human genome project. So first one is human genome contains 3164.7 million nucleotide base pair, and the total number of genes is about 30,000. An average gene consists of 3000 bases, but sizes vary. Largest known human gene, that is dystrophin on X chromosome, contains 2.4 million bases. So you can see DNA double helix, then you can see chromosomes, then you can see inside that DNA. Okay, inside DNA you can see nucleotides, and the right hand side you can see this dystrophin on X chromosome. So total genes involved here is 30,000, and the average gene consists of 3,000 bases. Again I repeat, total number of genes is about 30,000. Okay, so total number of genes are 30,000 and average gene consists of 3,000 bases. And dystrophin on X chromosome is the largest known human gene. So largest known human gene is dystrophin on X chromosome that contains 2.4 million bases pair or bases. 2.4 million bases. So 99.9 nucleotide bases are same in all people. Okay, so you should note 99.9 nucleotide bases are same in all people. Only 0.1, that is 3 into 10 to the power 6 base pair differences make every individual unique. Okay, so only 0.1, that is 3 into 10 to the power 6 base pair differences make every individual unique. And the functions of over 50% of discovered genes are unknown. Okay, so we should know that the functions of over 50% of discovered genes are unknown, and chromosome 1 has most gene, that is 2968 gene, and Y has least gene or a few gene, that is 231 genes. Less than 2% of the genome codes for protein. Okay, so less than 2% of the genome codes for protein. Next you can see very large portions of human genome is made of repetitive sequences. So there are traces. Okay, so there are traces of DNA sequences that are repeated many times. All right, so DNA sequences are repeated many times. So they have no direct coding functions. They shed light on chromosome structure, dynamics, and evolution. So about 1.4 million locations have single bases DNA differences. They are called SNPs. Okay, that is the full form of SNP is single nucleotide polymorphism, or SNPs. SNIPs. So this helps to find chromosomal locations for diseases. Okay, or for disease associated sequences and tracing human history. Okay, so thank you student, this was all about the entire video lecture. We have discussed about the topic Human Genome Project, that is HGP. This was from the chapter Molecular Basis of Inheritance, and here we could know that human chromosome, we could study the human chromosome, and inside the human chromosome we could study the genes and their functions. So thank you class. Again we'll meet in the next class. Thank you students.